You have a 1 minute.
1 minute?
Yeah. Really? Yeah.
I thought it's starting at 4:30.
No, no 15 minutes. So I think. Yeah. Yeah. Okay. Hello, everyone. Thanks for coming to the Kaiser Math Department colloquium. Let me introduce today's speaker, Professor Yuji Hirono. So Dr. Hirono got his PhD at the University of Tokyo in a physics major. And then he did a postdoctor at Stony Brook. And then he came to Korea as an assistant professor and group leader of Asia Pacific Center for Theoretical Physics in POSTECH. In fact, I listened his talk at the physics conference last year. Actually, I was really surprised when I listened his talk because he merged the topology and dynamical system and network theory. So actually, it was a most elegant and beautiful talk among all the talk I listened last year. So I invited him, and thankfully he got the acceptance. So hopefully you also really enjoy his talk. So let's welcome Dr. Yuji Hirono.
Yeah, thank you so much for the very nice introduction, and thank you all for coming here and having me here. I'm Yuji Hirono, and I'm from this Asia Pacific Center for Theoretical Physics located in Pohang, and today's talk is intended to be an invitation, to the study of the chemical reaction network from a topological perspective. And I'm especially happy to be able to talk to the people in the math department for two reasons. So one reason is that the analysis here involves certain mathematical concepts such as the homology group and so on. And the other reason is that I have an impression that I don't still get the complete understanding or picture of what is going on. And in order to reveal the whole structure, I believe that the mathematical viewpoint is going to be essential. So in the end of the talk, I'm going to raise a question so that you can think about and possibly solve in the future. And this talk is going to be sort of preparation to ask a question. So in the rest of this talk, I'm going to first give a generic introduction to the study of chemical reaction networks. And I'm going to introduce two analysis method in a similar split. So one is the sensitivity analysis, and the other one is basically the simplification of complex reaction networks. So let's start with this introduction. I believe that everybody knows what a chemical reaction is from your experience in the middle school or high school. So basically, a chemical reaction is a process to convert some chemical species to another kinds of chemical species, right? So for example, so here I'm showing you a chemical reaction, which is a combustion of ethanol. So we have this ethanol, which is combined with the oxygen, and they are turned into the carbon dioxide and water. So in this representation, the chemical species on the left are called reactants because they react, and those two are called products because they were produced. And a chemical reaction network is basically a chain of this kind of reactions. So there can be this reaction, and there can be some another reaction that uses the product of this, and there can be many chains of reactions. So basically, a chemical reaction network is a network made of reactions. And despite its name, I'd like to argue that this chemical reaction networks are quite ubiquitous object. So they certainly appear in chemical systems like chemical engineering or biological systems. But not only that, there are some other situations that can be described as a chemical reaction network. So the famous, what ecosystem is one example. And now that I think, many of you know this SIR model, which is a model based on, chemical reaction, and it models the spread of the virus. So this model assumes that there are only 3 kinds of people. So those who are susceptible to the virus, and those who are already infected, and those who are recovered from the virus. And I am one of those recovered person from the COVID. And the underlying reactions of this model are those 2 ones. So one process is the infection process. So when an infected person meets with a susceptible person, then this person can get infected. And another process is the recovery process. So one infected person can become recovered like this. And based on those reactions, we can write down the time evolution equation of this number of susceptible and number of infected and number of recovered people. So this, we can track the time evolution of those populations using this model. So another example in an ecosystem is the so-called Lotka-Volta model, so which describes the dynamics of the prey and the predators. So it assumes that there are only 2 kinds of species in this world, and let's call it as rabbits and wolves. And the underlying reactions are those 3. So there's a process of birth of a rabbit, and when wolf meets a rabbit, rabbit is eaten by a wolf, and that is going to contribute to the multiplication of the wolf. And there's also a death of the wolf process, and based on those reactions, we can write down the time evolution equation of those rabbits and wolves, and track what's going to happen in the system in the future. And even further, the chemical species in a chemical reaction can be some kind of a state. Then then we can describe various processes using chemical reaction network. For example, this represents the operation in a restaurant. So here is a customer 1 who entered this restaurant, and if waiter is free, then this take order reaction can happen. And then as a result, this customer is in the waiting state, and the waiter is in the order taking state. And this waiter is got to tell the kitchen the order of the customer. Then the waiter comes back to the free state again, and so on. So it can be used to model various processes in various situations in this way. So sure. It is a hypergraph. In the sense that, so in hypergraph we have vertices and hyperedges, and in this representation, the circles correspond to the vertices, and those rectangles correspond to the hyperedges. So in this sense, this CRN is almost equivalent to the hypergraph or Petri net, and it is a quite a generic framework to describe processes where some things are changing. And despite its name, it is more general than the usual directed network. So in the sense that a usual directed network is a special version of the chemical reaction network. So this is what a chemical reaction network is. And so one place where those chemical reactions play a fundamental role is inside living cells. So in the living cells, there are thousands of chemical reactions, and they form a pretty large network. And the function of this network are essential for us to be alive, because they are going to generate the physiological functions in the living cell. And one example of such function is metabolism, which is a process to convert what you eat into something useful for your body. And so this is here I'm showing you the metabolic pathway of E. coli, where we have this glucose, and there are many subsequent reactions like this. As you can see, this is rather complicated, right? And the analysis of such a system poses several challenges, which are both theoretical and also practical. First of all, the first difficulty is that available information is always limited, and this is especially true for the case of living cells. So, for example, we might not have the access to the dynamics itself. For example, we might not have access to the time evolution of this concentration or this concentration, and especially in the living cell, it's really difficult to track those quantities. And related difficulty is that it is really difficult to determine the reaction kinetics or the values of the parameters. So for each reaction, there's a reaction rate, which is the number of reactions happening per unit volume per unit time. And there's a parameter which is controlling the speed. And also there's something called the reaction kinetics, which specifies how a certain reaction rate, depends on the concentration of the reactants. So those two are needed if we are to describe the time evolution of the system, but we do not always have access to this information. And even further, the information of the network itself may not be complete. So there may be some reactions which we haven't discovered yet, behind this reaction network. And another problem is that the complexity of the system. So as you can see, this is a rather complicated system. And suppose that we are to run a numerical simulation of this system. Then you have to specify all the parameters for each reaction and all the reaction kinetics. So, and even if you succeeded in running this system and get certain behavior, you cannot be quite sure if this behavior is a fundamental property of this network or it is a result of the particular value of the parameter that you choose to run the simulation. So there are many difficulties, and we are in a kind of difficult situation. So how can we tackle this kind of problem? So I'd like to introduce an approach to the analysis of this system, which can be called as a structure-based approach. So it's something like this. So as we have seen, so there are two ingredients to specify a chemical reaction system. So one is the network structure. So that is this network structure. And the other information that we have to specify is the reaction kinetics and the parameter values. So if you look at these, the former information can be determined more robustly compared to the information of the reaction kinetics or parameters. So this information is model dependent and system specific information. And we can determine whether a certain reaction exists or not, more robustly compared to the question of whether a certain parameter takes certain value. So, and in this approach, we're going to base our analysis on the structural information of the network. And we do not make any assumption about the form of this reaction kinetics. So we are going to try to, exploit what we can learn from the structure itself. Not assuming the particular form of the reaction. So that is the basic idea behind this method. And I'd like to introduce two methods in this spirit. One is the sensitivity analysis, and the other one is the simplification of the reaction networks. And in the latter one, some mathematical concepts like homology groups and so on are found to be very useful.
before going into this method,
before going into this method, let me briefly, introduce how a chemical reaction system is described. So as I said earlier, a chemical reaction network is mathematically a directed hypergraph with weights. So it is directed because the reaction has a direction, and it has a weight because we have a weights here in front of those chemical species, and so it is a hypergraph, and the hypergraph it consists of it is a generalization of a usual graph, and it consists of vertices and hyper edges, and and a hyper edge in this graph can have multiple terminals. In the case of usual graph, an edge can have only two terminals, right? But hypergraph is a generalization of a usual graph where an edge can have multiple ends. And I think you can see that if you have this kind of reaction, it can be represented in a picture in this form. So where so basically the correspondence to the hypergraph is that those chemical species corresponds to the vertices. And I'm gonna represent this hyper edge by this rectangular box. So in this representation, chemical reactions corresponds to the hyper edges. And so given a reaction network, we're going to consider some dynamical system on that. And there are, different levels of descriptions, right? But so for example, when the number of the particles are large, then we can reliably use the deterministic, differential equation to describe the time evolution of the system. And in this case, the degrees of freedom that we are going to be dealing with is the chemical concentration of each chemical species, which are identified by BI. So this represents the chemical concentrations. And the other quantity is the reaction rate of a particular reaction that I denoted by EA, so which quantifies how many times a particular reaction occur per unit time and per unit volume. And for example, let's consider this kind of reaction network, which consists of 4 reactions. So there's a process of some input of V1 from outside wall, and there's also V2 is provided from the outside wall, and there's a reaction where V1 and V2 are combined into V3, and finally there's a reaction of the output of V3 to the outer wall. And I think it's natural. You can, the fact that the set of those reactions can be represented in this graph, is going to be, natural to you. And the time of, so we are interested in how those quantities evolve as a function of time. And its time evolution is governed by the ODE of this form, which can be written in this way. And so this just means that the concentration of V1 is going to increase if reaction 1 happens, and it's going to decrease if reaction 3 happens, right? So something like that. And it can be written, in a form where we can see the structure of the network. So if we write, in this form, and this matrix, so it can be written as a matrix times this vector of a reaction rate. And this matrix is called a stoichiometric matrix, which specifies how the network is connected. So for example, this reaction E1, if this happens, then the V1 is going to increase by 1. And when E2 occurs, the V2 is increased. When E3 occurs, then V1 and V2 is decreased by 1, and V3 is produced, and so on. And I think you can see that this is going to specify the structure of this network. And in addition, if we are to solve this equation, we have to represent those reaction rates as a function of the concentrations xi. And so there are, several. So this is a model-dependent part, and there are, several standard choices. And one, the most standard one would be the so-called mass action kinetics, where the reaction rate is, going to be proportional to the product of the concentration of the reactants. So in the case of this R3, its rate is going to be proportional to the concentration of the B1 and the B2 times certain constant. So the parameter appears in the specification of the reaction kinetics. So let me briefly remind you what I just said in a general setting. So in this description, deterministic description of chemical reaction system, the degree of freedom that we are going to be tracking is going to be the chemical concentrations of each species, and also the reaction rates, and the time evolution of those concentrations are governed by the rate equation. And on the right-hand side, we have two ingredients. One is this matrix called the stoichiometric matrix, and this characterizes the structure of the network, and this is the corresponds to the incidence matrix as a hypergraph. And the other ingredient is the reaction kinetics. So we are going to represent this rate as a function of the concentration of the reactions. And here some parameters entered. So this is a general structure, in this level of description. And now let's discuss the sensitivity analysis. So the basic idea behind this is something like this. So suppose that you encounter something very weird. Then what you do is to, for example, talk to it or touch it. So basically, you're going to try to perturb it and see how it responds to you, so that you can learn about this weird object. So this is the basic idea of the sensitivity analysis. And here we are interested in the sensitivity of the steady state. So if suppose that you have some chemical system, then if you wait long enough, then in many situations it reaches the kind of a steady state where no changes happen. And in terms of equations when the steady state is realized, the time derivative of this concentration is going to be 0. And by solving this equation, we can obtain the values of these concentrations and then reaction rate at the particular steady state. So now what we're going to do is to change the parameter. So we're going to change the value of a certain parameter a little bit, maybe. Then try to see how the steady state changes under this perturbation. And here, as I said earlier, we do not assume a specific form of the reaction kinetics. So let's try to do this. So for example, let's consider this kind of reaction network, which consists of 6 reactions. So there's an input of V1, and there's a process V1 to V2, V2 to V3, and so on. So this is the underlying network that we're going to consider. And suppose that we have waited long enough and system has reached a steady state. And let's try to perturb this system. So for example, we can change the parameter of E2. So, and what's going to happen here is that after change the parameter and wait for some time, then what's going to happen is that the concentration of B1 is decreased, but nothing changes for other parts. So what I mean by that is that the concentration of B2 changes. When I switched on the perturbation, but it comes back to exactly the same value. So that is what I mean by the fact that it's not changing. And if you instead perturb this parameter, E4, if you perturb this parameter for this direction 4, then so what's going to happen is something like this. So the concentration of B1, B2, and B4 are increased, and reaction rate of E2, 3, 4, 5 are increased, and that's basically it. And if you continue and, for example, modify the parameter, increase the parameter of E6, then the concentration of those quantities decreased, and also the reaction rates decreased, and that is the order change. So I think apparently, I mean, it shows a rather non-trivial pattern. I mean, I guess the perturbation, and we like to understand, you know, how this kind of a pattern, I mean, appears, and we like to understand its relation to the underlying network structure. So, and in order to do that, let me introduce some terminology. So we're going to be looking at subnetworks, which is going to be specified by the subset of the vertices, I mean the chemical species, and subset of the reactions. And we are going to say that a subnetwork gamma is output complete if there's a E gamma, which is the reactions included in the subnetwork, includes all the reactions which is affected by the chemical species inside this subnetwork. So what I mean is very simple. So one example of a subnetwork which is not output complete is this one, because the E3 reaction is affected by B2. So if we want to make this output complete, we also have to include E3. So that is what I mean. And then we're going to introduce some number, which is going to be a non-negative integer, and let's call it as an influence index. So for a given output complete subnetwork, which I denote by gamma, we want to define its influence index by this formula. So this is basically given by the minus the number of the chemical species inside the subnetwork, plus the number of reactions inside the subnetwork, minus the number of cycles, I mean, number of the loops that is supported in this subnetwork, plus, I mean, number of constant charges projected to this subnetwork. So it kind of looks like an Euler number, and importantly, this is determined according to the network structure, and it is insensitive to the, I mean, chemical reaction kinetics or the parameter values. And so those people have shown that this index is going to govern the response pattern of the system. So what they have shown is that, so if, I mean, you have some subnetwork, which is output complete, and if this index of this subnetwork is 0, then the change of the parameter of the reactions inside this subnetwork is going to be confined, I mean, inside this subnetwork, I mean, for the steady state. So if the index is 0, I mean, even if you change the parameters here, its effect is confined here and doesn't affect other parts of the network. And for this reason, a subnetwork which has a 0 influence index is called a buffering structure, because it works as some sort of a buffer to confine the effective perturbation. And the response pattern that we have I mean, looked at previously can be sort of explained, I mean, using this index. So you can compute the index, for example, for this part, and this has one species and one reaction, and no cycle and no constant charge. So this is index 0, and this part also has a 0 index. Here we have 3 species and 4 reactions, and there's a 1 cycle, and this part is also 0, and this, I mean, is consistent with the perturbation and response pattern that we have observed earlier. So by looking at this topological index given to a subnetwork, I mean, we can, I mean, estimate how far the perturbation propagates, and this is, I mean, determined by the structure of the network, and it's insensitive to the details of the system, like the parameter value or the reaction kinetics.
What was the parameter? The index, yeah, what was that?
This is, yeah, the number of conserved charge projected to the subnetwork. And in those examples, there's no conserved charge. So it is, 0 in all the cases.
You know, bidirection. Then we have one.
Yeah, I'm going to give you an example of a conserved charge in a chemical reaction system later. So we can get some insight from the existence of those buffering structures. So first of all, it's going to work as a kind of a biological firewall because the perturbation, the effective perturbation is confined inside the subnetwork. And often it shows this kind of hierarchy pattern. So there can be some buffering structure inside the buffering structure and so on. So this can be seen as a candidate of a mechanism to ensure the robustness of the system against the perturbations. And so we can also look at this kind of buffering structure as a biological unit in the sense that if this index is 0, this part can be controlled rather independently of other parts. And it also has some good property as a unit. For example, it is a buffering structure is closed under the intersection or union. Which one?
Yeah, close on it.
This can be proven. In fact, yeah, its proof is pretty interesting. I unfortunately omitted that slide, but it's very easy. And so let me mention one example where this kind of analysis was useful in interpreting the experimental result. So here we're gonna look at the metabolism of a plant, and that plant is called Arabidopsis thaliana. So this is not my work, and it's in Korean. It's called like this. I'm not sure what it. I'm not sure. Yes, and the process we are going to be looking at is basically the production of sucrose. So sucrose is a kind of a sugar, and it's an important ingredient in the plant. And so the authors has made a mutant which has the less ability to degrade something called pyrophosphate or PPI. So here I'm showing you the standard reaction network, which is responsible for the production of sucrose, and PPI appears here. And in those genetic mutants, this process is slowed down. It's not 0, but the reaction rate of those reactions are reduced. And so what they have observed is that, so indeed, since it has less ability to degrade the PPI, this mutant has more amount of PPI compared to the original one or wild type. And they have observed that the amount of sucrose produced in those cells is decreased for the mutant. So the question here is, how can we understand this kind of behavior or response of the system from it underlying the reaction network? And so yeah, this is what happened. And but if you look at this reaction network, and then if you change the parameter here and then decrease the parameter, what is can be shown is that it doesn't change the amount of sucrose produced here. And this can be explained by the fact that this part in fact is a buffering structure. So even if you change the parameter here, you cannot affect the other part. So then how can we explain this experimental result?
What actually is
yes, cross decreased like this. So the reason is that we should recall that this is only a part of the network and it's embedded in a larger network. And in order to explain this, so they have found that so this is the part that I have shown earlier, and they have shown that we also have to include those reactions as well. So that we can explain the fact that the reduction of this degradation rate of PPI leads to this decrease in screws. So this example tells us that we should be careful when extracting the part of the network, because if we do it inappropriately, then it can modify the behavior of the system. So this naturally leads to the our second topic, which is the reduction of a chemical reaction network. I'm not sure how I'm doing with time. Okay. So the motivation, to consider a reduction or simplification is something like this. So as we have seen, it's a complicated system, and it's difficult to sort of understand its behavior. And it will be very nice if we can sort of extract the important part of the structure for the relevant phenomenon. But if we do it poorly, it can destroy the feature of the original network. So the question here is how we can simplify a reaction network while preserving its important features. And so we have discussed this reduction problem, but originally our question was not practical but theoretical in the sense that this influence index is very nice, and it can be used to look at how the effect of perturbation propagates, but its appearance looks a bit mysterious, right? And I wanted to personally understand why it can be sensitive to the the pattern of influence inside this network. And so we have discussed this with a mathematician and with also a biologist. And what we have done is that we want to introduce a homology group associated with chemical reaction network, which is going to be a simple one. And using a certain elements of the homology group, we can decompose this influence index, in this way. And once we recognize that it can be represented in terms of some elements of homology groups, we can utilize the methods that are typically used in the study of those homological algebra. And what we have realized is that so there's a short exact sequence for a pair of chemical reaction network, and there's going to be associated the long exact sequence, and this can be seen as a mathematical representation of the process of reduction of a chemical reaction network, which I'm going to discuss a bit. So before discussing that, let me briefly, in one slide, remind you what a homology group is. So when we consider homology groups, we typically consider the chain complex, which is a sequence of some spaces. So it can be module or some vector space. And there's also some maps that is called the boundary maps for each space. And importantly, it goes to 0 when it's multiplied twice. And it is those spaces are labeled by some integer k, and an element of this C sub k is roughly speaking a k-dimensional object. And inside this k-dimensional object, there are some objects which goes to 0. When you take the boundary map, which means that there are some objects which doesn't have a boundary, And among them, some objects which can be written as a boundary of something. And the case homology group is defined by this. So it is the cycles, which are the object which doesn't have a boundary, modulo boundaries. So for example, I mean, if you look at this torus, and let's consider one-dimensional object, or which is basically a one-dimensional object which doesn't have a boundary, which is basically a cycle. And if you look at this one, this circle can be represented as a boundary of this disk. While if you look at this circle, there's no, disk which whose boundary is this circle. So this is one of the non-trivial elements of the homology group, and we can use this information to characterize the shape of some topological spaces. And so we are thinking about the chemical reactions networks, and one obvious, straightforward, very simple-minded way of defining a homology group is something like this. So we have, vertices and hyperedges as a constituent of a hypergraph or chemical reaction network, and we are going to consider the linear combination of the vertices with, for example, real coefficients. And let's call it as a zero chain. And we also consider the linear, combination of the edges. And let's call this vector space as a one chain. And in addition, we have to define a boundary map. And we are going to define a boundary action of the boundary map on its action on the basis of this space, and using the elements of the stoichiometric matrix. So this can be immediately understood if you look at the example. So the boundary map asks, so what is the boundary of a certain object? And if you apply the boundary map on this E1, it's going to give you minus V1, minus V2, plus V3. I think which is kind of natural and it makes sense to you. So this is the most straightforward and obvious way of defining the chains and also boundary maps. And higher chains are zero in this setting. So now we have a chain complex, which is rather small, but we can consider the homology groups associated with this chain complex. And so for each variable k, which is just 1 and 0, we can consider this kernel of this modulo, this image of this map. And we can so we have two homology groups, H0 and H1 for each chains, and we can identify those spaces as the following. So the space of H0 corresponds to the space of the conserved quantities in a chemical reaction system, and the space of H1 is going to be the space of the steady state reaction rate. So let's see some. And importantly, this information is going to characterize the steady state of a reaction system. So, for example, an element of H1 is called cycles, which might be slightly confusing, but anyway, so which is basically the closed loop made of reactions. So if you look at this, and there's one cycle here, and there's also another cycle here. Yes. So namely, the boundary is identified in this setting, and so those are the elements of H1, and the element of H0 corresponds to a certain linear combination of the concentration of the species that do not change as a function of time. So if you have, for example, this kind of reaction, V1 to V2, V2 to V3, V3 to V1, then the total of those species are going to be conserved because this number doesn't change I mean, and and this is going to give you an element of H0. And another example is something like this. So there's so there are 4 species, and there's a reaction to produce, I mean, those B3 and B4 from B1 to B2. And there's also an inverse reaction. And in this network, the B3 and B4 are always produced in pairs. So which means that the difference is going to be unchanged as a function of time. So this is also going to give you a non-trivial element of H0. And based on this idea of the homology groups, so similarly to the usual case, we can consider a subnetwork or subspace, and we can decompose this influence index using based on some particular component of the cycles and conserve the charges, and they in particular those quantities appear when we consider the long exact sequence associated with the pair of reaction networks that I'm going to discuss a bit later. So we now let's talk about the how to reduce or complicated reaction network. So based on this recognition of the homology group, so we have developed one method to systematically reduce the complicated reaction network to a smaller one. And this method consists of 2 steps. There are only 2 steps. So first of all, we are going to identify a subnetwork, which we want to eliminate. Then in the second step, we are going to actually eliminate this subnetwork to get a smaller network. And for each step, there are same problems to be considered. First of all, since we'd like to extract the important part, so we'd like to identify some parts which is unimportant. And so the question here is how we can identify such an unimportant structure in a big network. And another question is that, so suppose that we have chosen this subnetwork and decided to eliminate this, then if you naively just eliminate this, then the network is broken apart, and that is not quite what we want. So we sort of want to nicely match the input to the subnetwork and output from this subnetwork in a nice way. And the question here is how we can systematically so which means that we have to reconnect certain reactions. And the question here is how we can systematically perform this kind of a reconnection. And so I'm going to talk about the first part of identification of unimportant part later. And let me first talk about this elimination process of a subnetwork. So suppose that you have already chosen a subnetwork which you want to eliminate. And I'm going to denote this as a gamma, so which consists of the subset of the vertices, the chemical species, and subset of the reactions inside the whole network. And according to this distinction, we are going to separate the chemical species and chemical reactions to those that are inside the subnetwork and outside the subnetwork. I think this is kind of obvious and makes sense. And we'd like to eliminate this inside degrees of freedom. So in the reduced system, we should have this kind of time evolution equation. And here, the reduced variable should be the concentrations outside the network, the subnetwork that we have eliminated, because these are the remaining degrees of freedom. And we also have the reactions outside the subnetwork as a remaining degree of freedom. And so we have identified this X prime and R prime. And we also have to specify the connectivity of the network, which is specified by this stoichiometric matrix. And here, we are going to use, this form of the stoichiometric matrix. So first of all, this is the whole stoichiometric matrix, and we are going to divide it into 4 sectors according to whether this row is inside this subnetwork and outside, and also whether this column is inside direction in gamma and outside direction, and so on. So we have 4 vectors, and out of that, we are going to define the stoichiometric matrix of the reduced system in this form. So this is S22 minus S21 times S11 plus times S12. Here, this S11 plus is the Moore-Penrose inverse of the matrix S11, which is the generalization of a matrix inverse. And so there are several ways to see that this is a natural choice. So one is that if you perform this elimination of the internal degrees of freedom at the level of equation motion, this is the form that is going to appear. And we can also think about this way. So we can think about the mapping between chemical reaction networks. And in particular, we can think about a mapping which contracts subnetwork into a point. Then then we can show that the associated matrix representation of the stoichiometric matrix should take this form. And so, for example, if this is a whole network, and if you choose this subnetwork, then the resulting reduced network obtained by eliminating this is this one. I think this looks, reasonable and natural. And in a slightly, larger case, so this is the whole network, and I have chosen this as a subnetwork. And after elimination of this, we're going to get this network. And in this case, this is the whole network, and this is the subnetwork. And after elimination, we're going to get this network. So I'd like to emphasize here is that this operation is very, very easy because it just requires a matrix multiplication, which can be done very, very easily. So now let's discuss how to choose the part which can be eliminated and unimportant. So we have what we have shown in the current work is something like this. So suppose that there's a subnetwork, and this subnetwork is a buffering structure, meaning that the influence index is 0. Then what we have shown is that the corresponding reduced network, which is obtained by reducing this subnetwork, is going to have exactly the same steady state solution. For the remaining part of the network. So so what is so which means that a buffering structure can be eliminated safely, without altering the other part of the network. So what is nice about this is that this index is determined by only the structural information, and it is insensitive to the reaction details like the parameter values and reaction kinetics. So even if you don't know much about what's happening inside this subnetwork, you can safely eliminate this subnetwork as long as this topological condition is satisfied. So which means so because of this reason, it has a quite broad applicability. To the systems with generic kinetics. So let's see, apply this to a slightly complicated network. So this is the network that I've shown earlier, which is the metabolic pathway of the E. coli. And so there are like 17 buffering structures inside this network. And for example, we can eliminate this part, which results in this kind of network. And so there have been certain reconnections of the network. And we can continue this process. I mean, so after that, we can also eliminate, for example, this part, and and we can further eliminate this blue part as well to get this kind of network. And so we can perform this reduction sequentially, and the order of this reduction doesn't matter as long as the reduced part is a buffering structure. And we can see that the invariance of the steady state under those, under the reduction of the buffering structure. So here I have, compared the time evolution of chemical, concentrations that are still, remaining, and using the four, for those, four underlying reaction networks. And so this is the time evolution by each reaction network. And we see that there are some differences in the earlier part of the time evolution, but eventually they're going to reach, the same steady state value, which is ensured by the fact that the reduced part is buffering structure, meaning that the index is 0. 5 minutes. So, and so let me briefly mention how this result is derived. So as I said earlier, homology groups correspond to the steady state information of the network. And the reduction process can be represented as a short exact sequence of a chain complexes. So we have some subnetwork, which is inside of the whole network. Then we are going to eliminate this subnetwork. And so it can be implemented mathematically as a chain, a short exact sequence of chain complex. So this is the chain complex of the subnetwork and the whole network and the reduced network. And this is going to be a commutative diagram under a certain condition. And from that, we can derive the long exact sequence of the homology groups. So this using the standard method. And so which gives us the relation between the homology groups of the subnetwork and the whole network and the reduced network. And this gives us the constraint about the possible steady state solution. And this sequence plays an essential role in improving the invariance of the steady state under the reduction of the buffering structures. So basically, this is the end of my talk. So we have introduced one approach that can be called the structure-based approach, which utilizes the structural information of the network. And in order to do that, I think the fact that it can be, so, and this is determined by the only the structural information. And as such, it is closely related to the homology groups of defined out of the chemical reaction network. And finally, as a final comment, I feel like, there's some more underlying structure. So the question here is, how can we sort of categorify the influence index? So what I mean is that, so there are some systems, right, which is composed of some parts. And those parts are, interacting with each other. And I'd like to, so my question here is, what is the proper mathematical characterization of the relation between the parts? So if we can sort of, conceive some mathematical object that can represent the relation between the parts and how it is integrated as a whole, then I think that in that object, this influence index is governing the influence pattern. I believe that this index should naturally appear once we sort of identify the correct mathematical structure of the influences of parts. So that is, I mean, so that is basically it. So thank you so much for your attention.
So any questions? Yeah. So any questions?
Yes, so the entry is going to be in general. It can be non-integer, but if we start from an integer matrix, I mean, it is at least a rational number.
Between multiple S1 and S2 is it inverse could be rational, right?
Yeah, it's a rational number, and in general, the pure complement can get some rational number, so it can be non-integer.
So S2 and S1 can be multiplied. Does it always become integer?
Not necessarily. It can have a fractional number.
But S2 is always an integer number.
So yeah, I mean, this part contains some rational number.
S1 and S1 are multiplied.
Yeah. I think that is not guaranteed. Yeah, in general, this is going to be a rational method.
Can we reduce the model? And it's the rational to reduce the model.
That's right. I mean, but so in that sense, the reduced system is not strictly the usual, chemical reaction system. It is a dynamical system which can reproduce the steady state solution of the original system. I think I had one, but yeah, let me think about that. Yeah.
Any other questions? Yeah. Yeah, you look like you.
That it can be possible that prime can have fractional. So when you make another quickly graph out of that S prime, you might have some 0.5 times some n, 0.7 times some. So you can really make homological concept out of that. Coefficient group should be not integer, but rational.
Yeah, so when the coefficient is a rational number, we consider linear combination with a real coefficient.
You're forward here.
Yeah.
When you consider the real coefficient, what exactly are those coefficients mean?
Those coefficients?
Just coefficient.
Yeah, I think at this level it is formal combination. But I think that
at this level it's just
yeah, so but the cohomology group has a more intuitive meaning. So I mean, if we can consider the map from the space of chains to a real number, then I mean that can be interpreted. For example, in the case of a map from zero chain to a number, we can interpret it as a concentration. And also the cohomology H1 is going to be, or C1, I mean, is going to, the cochain, the element of cochain can be regarded as the, I mean, value of the reaction rate, I mean, associated with certain reactions.
Fire question? Okay, let's thank the speaker. Recording 거 어떻게 보낼 수 있어요?
How is this different from, so you're only doing
저장이 될 텐데 그렇죠. 종료를 하면 아마 스탑이 될 거고.
when you're only doing
기록 중지.
zero and one.
네, 그거 맞고.
Like this.
아, 네, 종료하면 바로
기록 중지하고
I
종료하면 돼요.
don't see why